Welcome to the pre-lab discussion for experiment 1, thin layer chromatography. At the end of this experiment, you should be able to define mobile and stationary phase, determine the RF of a molecule, such as the active pharmaceuticals you're going to study in lab this week, predict the effect of changing mobile phase on the RF of a molecule, and determine the relative RFs you would expect for two molecules based on their structure. First, let's start out by defining TLC, or thin layer chromatography. This is an analytical technique which is used to separate small quantities of a mixture. Depending on the number of components or spots you see on your TLC plate, you can determine the purity of the mixture. By comparing your sample to a known reference compound, you can determine the identity of a sample. And later on in your organic chemistry career, you will be able to monitor the progress of chemical reactions using TLC. So the picture shown here is a TLC plate under a UV visualization lamp. It's important to understand that the TLC plates we'll be using in lab are coated with a white powder known as silica gel. And the most important piece of information I can give you about silica gel is that it's very, very polar and it's inorganic. It is more polar than most of the organic solvents that we would use in the lab. This coating is called a stationary phase because during this technique, the powder never moves. It stays adhered to the TLC plate. So when we want to use this plate for analysis of samples and reaction mixtures, we place the plate into a developing chamber, which in our lab will be a beaker. This is often covered with a watch glass to prevent evaporation of the solvent and help saturate the environment within the beaker with the solvent vapor. The stationary phase, as we mentioned, is the silica gel coating. And inside of the beaker, we place an organic solvent, which is known as the mobile phase. This is because this solvent will travel up the plate. And as it does so, it will interact with the compounds you have spotted on the plate. So in order to carry out a TLC analysis, you're going to begin with your TLC plate. And outside of the chamber, you're going to gently mark it with a pencil and spot a solution of your sample using a small glass capillary tube. We'll cover the precise technique for this during our pre-lab discussion in the lab. Before you place your plate into the beaker, you're going to add your eluting solvent or mobile phase into the developing chamber, just a small amount. You don't actually want it to come above the line you've drawn on the plate where your sample is originating. And then as you place your plate in and cover the beaker, the eluting solvent, that mobile phase, will travel up the plate and different components of your mixture will separate depending on their different affinities for the solid stationary phase and the liquid mobile phase. So to understand why different compounds will travel different distances up the plate, it's important to look at their structure, and you're beginning to develop an understanding of organic structure in the lecture. So what is most important in this type of chromatography is the polarity of the molecules. So the silica gel itself is very, very polar. It's actually a silicon oxide surface shown here. And because of all of these OH groups, it's really, really polar. So it turns out that polar organic molecules will adsorb onto the stationary phase with stronger intermolecular forces. As the solvent travels up, this polar molecule will spend time adhering to the solid and a smaller amount of time actually traveling up the plate with the mobile phase. So very polar molecules are often expected to travel not as far up the plate compared to nonpolar molecules. So to put this into action, let's take a look at the molecules A and B here. So based on your knowledge of organic chemistry, ask yourself, which one you expect to be polar relative to the other. And hopefully you've recognized that molecule B, which contains this carboxylic acid functional group, should be the more polar molecule. Molecule A is a simple hydrocarbon. It doesn't contain any hydrogen bonding functional groups. Given that A is the less polar compound, we expect that in this equilibrium between our polar stationary phase and our less polar solvent, that A will spend a greater amount of time traveling with the solvent, while B will adhere or adsorb more tightly to the solid phase and will travel less, spending less time with that mobile phase, leading to a lower RF spot. We cannot predict absolute RF values. They must be experimentally determined, and they largely depend on the particular silica gel plates you're using and the mobile phase you have selected. However, we can reliably predict the relative RF values of compounds such as we've done for A and B. The designation RF is used to describe how far a spot has traveled up the plate. RF here stands for ratio to the front. So the way an RF is calculated is you define the origin of your compound or your sample as the pencil line where you initially spotted your solution. The mobile phase is allowed to travel a certain distance up the plate. We usually stop it before it hits the very top. The distance the mobile phase has traveled is known as the solvent front. When you visualize your plate, you'll see spots that represent different compounds in your samples. You typically go to the middle of the spot, and the distance that center of the spot is from the origin, denoted here with B, is the distance your compound has traveled. If you divide this by the distance of the solvent front, you can obtain the RF value. And oftentimes these will be reported in literature, especially if you're studying novel organic molecules. So now that we understand how to describe how far up the plate a molecule has traveled, and we understand how to predict the relative distance two molecules will travel, there's one more parameter that we're going to study in the lab this week, and it's to look at the effect of solvent polarity on RF values. So consider the example we did before. Let's say we've started with a 50-50 mixture of hexanes and ethyl acetate. So that's the mobile phase that has been placed into our developing chamber. And to remind ourselves, as we're getting into naming organic compounds in lecture, hexanes refers to a 6-carbon hydrocarbon chain, 1, 2, 3, 4, 5, 6. No polar functional groups at all, simply hydrogen and carbon. So this is a relatively non-polar solvent. And ethyl acetate, we'll show in blue here, contains the functional group known as the ester. And you can see that because we have begun to place oxygen into the molecule, and we have this carbon-oxygen pi bond, this molecule is more polar than hexanes. So this is a slightly more polar component of our solvent. Remember, both of these solvents are much less polar than the silica gel itself. So as they travel up the plate, again, the only thing that will dictate the relative RF values of A and B is that A, the less polar compound, will travel more with the organic solvent, and B, because of this very polar carboxylic acid, will spend more time adsorbed onto the silica gel surface. So we decided in the previous slide that this would represent the relative RFs of A and B. However, if we switch the mobile phase inside of our developing chamber, and now we switch it to 100% ethyl acetate, as we increase the polarity of the eluting solvent or the mobile phase, we say that the eluting power increases. What that means is that all organic molecules will spend more time traveling with the mobile phase than they did in the relatively less polar solvent. So A will still travel farther because, again, it less prefers the silica gel surface overall, but it will travel more than it did in the plate above. B will still travel less than A, but we might expect its RF to increase. And you and your partner in lab will choose two different solvents and study the same compound and its elution in those solvents to really observe and confirm that what I'm telling you here is accurate. So as a take-home message, a couple of important things when predicting what you expect to see on a TLC plate. As the polarity of eluting solvent increases, its power to move molecule increases. And so the RF of all organic molecules will go up relative to conditions where a less polar mobile phase was used. When spotting two compounds, we expect that more polar molecule to interact more strongly with the polar silica gel, and thus it will spend more time adhering to that surface, and it will not travel as far up the plate. Now that we understand the background of TLC, I'm just going to briefly explain how we're going to use this in lab this week. So you're going to be given or choose 5 unknown samples. These samples correlate to known over-the-counter analgesics. And what you're going to be doing is you're going to be analyzing them by TLC to figure out how many active organic components each of the unknowns has. And then you're also going to study some reference compounds, such as acetaminophen, caffeine, ingredients that are commonly placed into these analgesics. By matching the RF of the spots in your unknown to the spots of these known organic compounds, you can say, for instance, my first unknown sample appears to have acetaminophen and caffeine, whereas my second unknown sample only has caffeine. Okay. And the number of spots will correlate to the number of components in your sample. And these RF values will really allow you to learn something about what specifically is making up each of the painkillers that you're looking at. So the commercially available samples are anisin, Bufferin, Excedrin, Tylenol, and Motrin. And this chart from your lab manual tells you which active ingredients are present in each of these over-the-counter pills. So for instance, if you do observe a spot that matches the RF for caffeine, your sample cannot be Bufferin because Bufferin does not contain caffeine, nor can it be Tylenol or Motrin. So this is how you're going to go about matching the unknowns you've been given to one of these common analgesics that most of us have taken before. Lastly, we keep talking about these spots we're going to see on the TLC plate. What you'll notice about your samples is that they're typically clear and colorless. So you might be wondering, how am I going to see these spots? When we buy the silica gel plates, they're coated with a fluorescent material. And when UV light shines on a fluorescent material, it gives us green light back. So everywhere on the plate that none of your organic molecules are, you'll see the green light come back at you. If you had an organic molecule that absorbs UV light, instead, you'll see sort of the absence of green light, which may almost look like a dark red spot. And that's how we know we have one of these important components we're looking for. Again, we'll talk about the specific experimental details, such as how to handle your TLC plate and how much liquid you want to spot at the point of origin during our in-lab discussion. But as a careful reminder, it's very important when you're using an analytical technique to determine how many compounds are present in a sample, that you use a clean TLC spotter for every new spot you make, and that you don't double dip or cross dip a spotter between samples. TLC is pretty sensitive. You only need a tiny bit of material. So if your spotter is even a little bit contaminated, it can lead to false positive results. Go ahead and read the lab manual that corresponds with this, and I look forward to seeing you in lab this week.